Okay, great. Well, thanks for coming to visit me, and thanks for the opportunity to talk to you all today. It was a very short drive for me this morning to show up. But I wear a few hats here at UD. One of them is as director of the Bioinformatics Core, which happens to be located directly next door to the sequencing core that Bruce Kingham talked to you about yesterday. So we've been on this PacBio train with them from the beginning since they got the RS in whatever that was, 2010, 2011. So we've been working with PacBio data for a long time. And having multiple hats, the temptation to move across my hats to apply technologies is always there. So I really have been looking for a while at good ways to use that in my own research program. And I look at viruses, and when I talk about viruses, what I'm talking about are microbial viruses, which are the vast majority of viruses out there. Mainly bacteriophage, some archaeal viruses, but pretty much anything that'll infect a microbe. And if you look at just double-stranded DNA viruses, there are 10 to the 31 of those guys estimated out there in the world in various environments, which is an astronomically huge number. It's hard to grasp, but it tends to be 10 to 1,000 times the concentration of their hosts when you're looking at them. And really, in these microbial populations, which I think today we can appreciate that we live in a microbial world, there are microbes everywhere from your gut to sediments miles below the bottom of the ocean that are controlling things ranging from our health to biogeographical cycles, or geological cycles that control carbon and nitrogen on the planet. And really, these viruses are acting like puppet masters. They control those populations, they keep them in check, and they shuttle diversity around between them, not only between the viruses, but also between the hosts. But despite their importance, they're really poorly understood. It's really tough to study these guys. And one of those reasons is because we don't have a universal genetic marker that we can go out there and look at all viruses and see how they're related to each other and be able to census them in the environment. There's no 16S equivalent, 18S equivalent. So it makes our job much more challenging. Obviously, these guys are really small. We, most of the time, don't know what they are, so we certainly don't know how to culture the host that we'd have to work on them in the lab with. So we've had to look at other methods, and shotgun metagenomics has really been a godsend for viral study, but at the same time, it's also been a curse because it is really, really hard to deal with viral metagenomes. From building them to analyzing them, the whole process is very difficult. So one of the big, looks like we have a little bit of a title issue here. One of the big issues we have with this is functional assignment is very difficult. We know some viral genes, what they do, just from homology to known genes. But even in cases when there is homologous function, sometimes viruses are so distant that you do not see that homology when you're looking at the sequence level. So assigning that can be very difficult. So when you're looking at a soup of unknown DNA sequences, you really have trouble saying what is actually there sometimes. And this is kind of an old graph from quite a while ago, but the point here is the ones that have color to them on this bar graph are the most predominant protein clusters in an aquatic environment we were looking at. And of those, only 38 of the top 100 had been seen before. And of those, all the ones with arrows pointing to them, most of them were uncharacterized protein, right? So even though we'd seen it before, we didn't know what it did. And this still rings true today. The numbers may be slightly different. But the reasons for this, our database is incomplete. Like I said, culturing these and actually doing lab experiments, very difficult. And the way that we analyze metagenomes has up until now tended to be very ORF-centric. We look at a single protein sequence that we can kind of divine out of this metagenome. But we haven't always been able to get good assemblies where we can see what the whole genomic context of that is. Coming up on that, you would really think with viruses, they have short genomes. The ones we look at, they tend to range from, say, 30 kb on the very small side to maybe 200 kb if there's fairly large ones for bacteriophage. They tend to be highly coding. They don't have a lot of repeat regions. So you'd think short read technology would be great for doing those assemblies, right? And the answer is yes, if you've got an isolate and you're sequencing an individual genome. But when you're looking in an environment with a soup of these, then it's very difficult. You see lots of overlap between the diversity. There's microdiversity going on where you see variants at the SNP level, but there's also much larger genomic rearrangements going on with cassettes coming and going. So when you try to do assemblies of these, this fast evolution that viruses have in an environment mean that there's not a single type for that virus. There's a continuum of genetic diversity for it. You see gene swapping occurring between both like viruses and fairly divergent viruses. And really what that leads to is when you do a short read assembly of a virome, you end up with a tangled mess of things that have maybe something in common, but often can become chimeric, or they become truncated earlier than you'd think they should. So just to illustrate how we can see this, just imagine this as a graph showing how the assembly for a very short genome went together. It's PHY-X174. And there's two different variants in this population. And you can see, pushing the wrong one. There we go. So you can see this is kind of the circle of the genome, and at these points, we've got a few branches where there happen to be a few genetic variants. That's how you would expect one of these to look. But when we look in a metagenome, we see things a little bit more like this, where there's just this tangled mess of overlapping sequence. And we see them over and over again within a metagenome. And occasionally we'll even see messes like this that we have no idea what to do with it. So the question that we asked was whether PacBio long-read sequences can help us to disentangle this mess a little bit, to help us make more sense of it. And the short answer is yes, it has definitely helped us. Getting enough DNA from viruses, very tricky. Getting a quality viral metagenome, very tricky. So getting the technology to work has been a major limitation. I can tell you on a recent project we did, we collected 120 liters of seawater, and we concentrated that down to about 20 milliliters of viral concentrate at the end. It's an incredible amount of work just to try to get enough material so that we can go forward with these types of metagenomes. And just to show you it works, this is kind of just an observational thing, but when you think about kind of the classic assembly metrics like N50, they don't necessarily describe the types of improvements we see really well because viruses are short genomes to begin with. So you don't see those great leaps in contig length. But what we do see are when we observe genes that we're interested in within these populations, when we've used PacBio, this is a single sample we collected about 70 miles south of here in the Chesapeake, looking at seawater. And we're interested in this DNA, I keep clicking the wrong button, there we go, DNA polymerase 1. If we look for full copies of that in our single metagenome, that was a hybrid Illumina PacBio assembly, we found about 2,000 within that collection. If we were to look at 79 other aquatic libraries that we were holding on to at that point, been assembled by various different methods, we found only about 1,200. So we're seeing a lot more. Is that sample richer in DNA pol A than other samples? Probably not. What we think we're seeing is that we're actually able to disentangle these variants of viruses that would have gotten flattened in a short read assembly. We're also getting full length, so we get less fragmentation within them. So why DNA pol A? Why did I pull that out as an example? It's a gene we've been interested in for a while. We've had students working on it. There is no universal marker for bacteriophage, but we do find this in about 25% of double-stranded DNA bacteriophage. So it's a pretty common marker. And we've also discovered something about it several years ago, that there's a single amino acid residue within that protein. It's at position 762 in E. coli. And it's been known for quite a while that if it has one of 3 different amino acids, it will affect the accuracy and the speed of the enzyme. And doing observational work, looking at different viruses, we've discovered that this can actually tell us whether it's a lytic lifestyle virus or a lysogenic lifestyle. So lytic comes in, immediately lyses the cell. Lysogenic incorporates itself in the genome, sticks around a while. So why does that matter? Think about lytic viruses quickly replicating themselves. They can make tons and tons of copies, errors in a few. That's okay for them. Maybe we'll have a variant. Maybe we'll have something really special come up one day. But by selecting that polymerase that makes an error, they've actually increased the rate of evolution. So they can make more copies of themselves, being more divergent. With these temperate phage, they need to be more in sync with their host, so it actually makes sense that they don't want to have that rate of evolution be quite as high. So other ways that these long reads are helping us is now that we can not only look at our DNA pol A and see this mutation, but we can also see the genes around it, and we can see them coming and going in the population, and we can tell that they actually also impact that viral phenotype, that way that it replicates, the speed that it can possibly replicate with, the number that it may be able to produce in a single burst event. So this is helping us to go through and take rules for genes that tend to occur together with various variants and make a rule book for how viruses behave based on their genomic content, which is an ongoing project that we're expanding on currently. And I think I'll skip over this a little bit, but just to mention, we're also using this data in a slightly different way, looking at genes that occur together, so guilt by association, and using that to help us assign putative functions to genes that we see over and over in these metagenomes in association with other genes. So lots more to come there. All right, I'll end it there.
That's amazing.